Insulin, discovered in 1921 and initially sold for $1 per dose, is now priced over $300 per vial in the United States. This exponential increase is not driven by manufacturing costs, which remain minimal, but by corporate control over intellectual property. The three major manufacturers, Eli Lilly, Novo Nordisk, and Sanofi, control 90% of the global market, engaging in a practice known as evergreening, where minor modifications to existing formulations secure new patents, extending monopoly protection indefinitely. This system transforms a basic biological necessity into a commodity accessible only to those who can afford it, leading directly to preventable deaths among diabetics who ration their supply. The core technology for producing human insulin, recombinant DNA insertion into E. coli bacteria, has been stable and widely understood since the 1970s. The scientific knowledge is public domain. The restriction is purely legal and economic, enforced by the state apparatus protecting corporate profit over human life. This manufactured scarcity is the fundamental problem that decentralized, open-source biology seeks to dismantle. Intellectual property, specifically pharmaceutical patents, functions as a state-backed monopoly, fundamentally contradicting the free flow of information and technology necessary for human well-being. Anarchist analysis views patents not as protection for innovation, but as a mechanism of violence, using the coercive power of the state to restrict access to life-saving knowledge. The initial patents on insulin expired decades ago, yet the manufacturers maintain control through complex legal maneuvering around delivery systems and minor molecular variants. This legal framework ensures that generic competition is blocked, maintaining artificially high prices and guaranteeing massive profit extraction from the sick. The system prioritizes the abstract right of ownership over the concrete need for survival. Challenging this monopoly requires direct action that bypasses the state and corporate structures entirely, focusing on decentralized production and distribution based on mutual aid principles. We must recognize that the knowledge required to produce insulin belongs to humanity, not to shareholders. Producing human insulin relies on established synthetic biology techniques. The process begins by synthesizing the gene sequence encoding the human proinsulin protein. This sequence is then inserted into a plasmid, a small circular piece of DNA, which acts as a vector. This recombinant plasmid is introduced into a host organism, typically the bacterium E. coli or baker's yeast, Saccharomyces cerevisiae. These microorganisms are then cultured in large fermentation tanks, where they rapidly multiply, expressing the human proinsulin protein. After fermentation, the proinsulin must be purified and cleaved to form active insulin. The purification stage is the most technically demanding part, requiring chromatography to separate the insulin from bacterial proteins and other contaminants. While the basic steps are standardized, achieving pharmaceutical-grade purity requires rigorous quality control protocols. The key insight is that the necessary biological tools and protocols are accessible to community labs, provided they adhere to strict safety and quality standards, effectively democratizing the means of production. The Open Insulin Project is a decentralized initiative dedicated to developing and publishing open-source protocols for insulin production. Their goal is to create a generic, non-patented insulin strain and share the complete manufacturing blueprint, including purification and quality assurance methods, free from intellectual property restrictions. This project operates on the principle of direct action, creating the alternative rather than lobbying the existing power structure. The project focuses on using publicly available information and non-proprietary strains to ensure the final product is legally unencumbered. By making the entire process transparent and peer-reviewed, they aim to establish trust and safety outside of traditional corporate secrecy. The resulting protocols allow community biolabs, universities, or small nonprofit manufacturers to produce affordable insulin locally. This approach fundamentally challenges the centralized hierarchical model of pharmaceutical supply by establishing horizontal, resilient production networks.
Community biohacking labs, such as BioCurious or GenSpace, serve as the physical infrastructure for decentralized pharmaceutical production. These spaces provide shared equipment, including fermenters, centrifuges, and chromatography columns, making complex synthetic biology accessible to non-corporate researchers and activists. These labs embody the anarchist principle of prefigurative politics, building the desired future society in the present by creating autonomous, non-hierarchical institutions. Instead of relying on centralized government regulation, safety and quality control in these environments are managed through rigorous, transparent peer review and community accountability. The shift from centralized proprietary research to decentralized open collaboration accelerates innovation and drastically reduces the cost barrier. If the means of production are distributed and the knowledge is free, the ability of corporations to enforce scarcity collapses. This model transforms patients from passive consumers into active participants in their own healthcare provision. Producing safe injectable insulin requires stringent quality control, particularly concerning purity and potency. The primary risk in decentralized production is contamination by bacterial endotoxins or incomplete purification, which can cause severe adverse reactions. Open-source protocols must incorporate detailed steps for high-performance liquid chromatography and mass spectrometry to verify the final product's identity and purity. Community labs must establish transparent, auditable testing procedures, often collaborating with university labs or independent testing facilities. This reliance on peer verification and open data replaces the opaque proprietary testing methods used by major pharmaceutical companies. Safety is achieved through radical transparency and mutual oversight, demonstrating that effective regulation can be horizontal and community-driven rather than vertical and state-imposed. This commitment to verifiable quality is essential for building trust and ensuring the viability of autonomous healthcare solutions. The open-source insulin movement is a practical application of mutual aid, where individuals and communities cooperate to meet their needs without relying on hierarchical institutions or market mechanisms. Mutual aid is distinct from charity. It is a reciprocal relationship based on solidarity and the recognition that collective well-being is essential for individual survival. In the context of healthcare, this means replacing the profit motive with the imperative of care. When communities produce their own essential medicines, they remove the leverage corporations hold over their lives. This form of direct action bypasses the political process, which is often captured by corporate interests, and immediately addresses the material needs of the population. The goal is not to reform the existing pharmaceutical system, but to render it obsolete by creating functional, accessible alternatives rooted in decentralized cooperation and shared resources. Operating outside the established regulatory framework, such as the Food and Drug Administration, presents significant legal and logistical challenges. Producing and distributing medicine without FDA approval is illegal under current law, designed to enforce the centralized control of pharmaceuticals. Open-source projects navigate this by focusing on research and development, publishing protocols, and potentially operating in jurisdictions with more permissive regulations regarding non-commercial, community-based production. The long-term strategy involves creating a parallel system of certification and quality assurance based on open standards, challenging the legitimacy of state-mandated monopolies. Legal defense and political organizing are necessary components to protect researchers and distributors engaging in this form of medical disobedience. The ultimate goal is to establish the ethical imperative that access to life-saving medicine supersedes intellectual property law. The success of open-source insulin provides a scalable model for addressing monopolies across the entire pharmaceutical landscape. Many essential medicines, including certain antibiotics, epinephrine, and HIV treatments, rely on technologies that are decades old and easily reproducible. Applying the open-source, decentralized model to these drugs could drastically reduce global healthcare costs and increase access in underserved communities. This movement represents a fundamental shift in how society views scientific knowledge and technology, from proprietary assets controlled by a few to shared resources managed by the many. The future of healthcare lies in horizontal networks of biolabs, transparent protocols, and community-driven quality assurance, ensuring that medical necessity, not profit, dictates availability. This is the practical realization of anarchist principles applied to the most critical area of human existence,